I'm Kazuhito Tabata from University of Tokyo. I'd like to begin by thanking the organizers for this opportunity. Today, I will describe a special research project sponsored by Japan Cabinet Office. So first, I'd like to introduce IMPACT. IMPACT is an initiative of Japan Cabinet Office. The project supports researchers with a high-risk, high-reward project. Currently, IMPACT found 16 program managers. The program manager of the project to which I am affiliated is Hiroyuki Noji. Our ultimate aim is the creation of the artificial cell using the microreactor array here. The project can be broken into the 3 sub-projects. In measure, we aim to enhance the detectability of single molecule reaction, like ELISA, through new quantitative and sorry. Sorry. Oh. Oh, okay, sorry. And high resolution technologies in create we aim to synthesize functional protein by directed evolution from single molecule DNA. Finally, in proliferate, we aim to reconstruct fundamental cellular function such as cell division and propagation inside our reactors. Today, my attention will be on the proliferate project. The work I will describe is managed by 2 people, Masayuki Suetsugu and it's myself. Dr. Suetsugu worked with exponentially amplifying circular DNA like a bacterial genome. He achieved this using the cell-free cloning system in which he reconstituted E. coli genomic DNA replication system in vitro. So my work involves creation of the artificial cell by fusing together the content of bacteria and microdevice. The first topic is here. So contemporary gene cloning method involves the transformation of the plasmid and culturing the cell. So this process normally takes 2 to 3 days. Moreover, there are a number of biohazard includes. In our method, we can extract required genome replication component and for E. coli. So furthermore, we develop methods that combine the gene fragment assembly lead to the cell-free cloning system. This approach reduces exposure biohazards, also because we demonstrated it using the E. coli genome replication system. We proved that it can handle the large DNA. So in this slide, I give an explanation of the DNA replication and amplification using our in vitro system. We named this process replication cycle reaction, RCR. The reaction involves the minimum component of the gene replication. The replication and amplification is achieved by repeating the cycle of duplication and amplification of circular DNA with the Orishi region. Here is the result of RCR experiment. This graph shows the 10 kilobase Orishi plasmid amplified using RCR. So after 100 minutes, we see a single plasmid was amplified 1 billion times. The protocol is amplification is because of limited substrate. To prove this, we took part of the reaction solution and moved to a new reaction solution like this, which lead to further amplification. Finally, the error rate is very low. 1.4 10 power minus 8 error per base per cycle. And here, I show the amplification of the large DNA by RCR. It is possible to amplify DNA 200 kilobase in size. So in the lab, we have achieved 350 kilobase and are now working on the DNA megabase in size. In this slide, I put together our gene assembly method and RCR for in vitro cloning. First, 21 DNA fragments are ligated, and then RCR is applied for the amplification. The electrophoresis image confirms the ligation product like this. To the right, you can see the RCR amplification product here. So we can see in vitro cloning was possible based on the supercoiled DNA. Another point about RCR is that only circular DNA can be used as a substrate, and that it can amplify many circular DNA molecules. Therefore, even if the efficiency of the ligation reaction is low, cloning can be quite high. Furthermore, genes that normally cannot be cloned can be cloned with our in vitro system. Ultimately, we expect in the future to clone the entire genome of large bacteria DNA. If you think this system could you help your research, please know that we have prepared an in vitro cloning kit. So this kit will be available free for this summer. Please contact me if you are interested. So next, I'd like to introduce my research. In this research, I fused together bacteria and a microreactor of the approximately equal volume to create the artificial cell. Here is a photo of our micro device. In the experiment, I introduced the bacterial genome and for the lipid bilayer is sealing with a lead. E. coli is integrated into the reactor by membrane fusion. The resulting hybrid cell has all component of the E. coli cell, which the lipid membrane contains all membrane proteins. The hybrid cell show the evidence of the growing and dividing, the ability to create a new bacteria. To observe the properties I mentioned in the previous slide, we investigated arrayed lipid bilayer chamber named ALBIC. So we introduce a fluorescent dye like this. If membrane barrier fails, then the dye escapes from the chamber and no fluorescent signal is observed. But we can clearly see fluorescence confirming the membrane bilayer. Next, we confirmed that bilayer membrane had proper function. This time, we observed alpha hemolysin. We can evaluate alpha hemolysin function by diffusion of the fluorescent dye. Here you can see the fluorescence signal decreasing with time, like this. Based on these findings, we confirm that our ALBIC does not impair the membrane bilayer. In this slide, I describe the fusion between the E. coli and ALBIC. I'd like to show you a video of the fusion. In E. coli have a GFP fluorescent. Once fusion occurs, size of the green light matches the size of the microchamber, like this. Please take a look, this and this and this E. coli. Again, moment green light matches the size of the chamber, we confirm the creation of the hybrid cell, like this. And this graph shows the fluorescence intensity of the hybrid cell. Each point on the graph represents a single reactor. The x-axis is represents fluorescence the beginning of the experiment. The y-axis represents fluorescence 3 hours later. The higher point, the more GFP was synthesized. You can see the chloramphenicol reduce the GFP expression. On the other hand, ATP only had effect on the GFP expression, but variance increased. Okay, restart. In this graph indicating the GFP was synthesized some hybrid cell. In this slide, I describe the beta-galactosidase expression in the hybrid cell. We introduce a beta-galactosidase plasmid into the reactor and measured its expression by observing the fluorogenic probe spider. So this movie showed a change in spider fluorescence, which confirmed beta-galactosidase expression in a hybrid cell. So the fluorescence could also inform us how many molecules of beta-galactosidase were synthesized. We found an average of 100 monomers per hybrid cell. These results demonstrate central dogma applied to our hybrid cell. Finally, although rare, we found evidence that hybrid cell could change its morphology and divide. Hybrid cell can be seen in the center of this movie. You can see the division, and here you see the hybrid cell elongate and engage with the reactor, like this. In this red circle, you can see the growth-like objects. Like this, and here. Finally, in this movie, you can see the small particle in the inside of the hybrid cell, like broccolis. These observations demonstrate E. coli regeneration in the microdevice. To conclude, I'd like to summarize my presentation. The most important point is the in vitro cloning kit. Thank you for your attention.
Thanks. Thanks. It was very interesting.
Thank you.
I'm wondering about the in vitro cloning kit and whether it could replace like in vivo cloning. So have you shown that you could go to limiting dilution of single plasmids and get clonal amplification of a single plasmid?
Yes. Yes.
One of the advantages of in vivo cloning is that you get one plasmid per cell, right?
Mm-hmm.
And so is it hard to avoid single molecule contamination in that setting?
Contamination is a problem, but so this system needs a ORC region. So without ORC region cannot amplify. So if ORC region contaminated plasmid plasmid have a ORC region, it's amplified. But I think I think it's difficult to remove the contamination.
In the case where you're using the cell with the bioreactor,
Yeah.
your supply of ribosomes, etc., is entirely coming from the incoming cell.
Mm-hmm.
What's the difference in volume? What's the difference in volume between the incoming E. coli cell and your bioreactor? Because you must be getting a big dilution, and it's surprising that you get as much expression and so on, and that's obviously going to limit the ability to produce the hybrid cells that you want in the end.
Yeah, very good question. So this my bioreactor is very tiny, as same as bacteria volume. So in my case, so E. coli size is about 3 or 4 micrometer diameter, about 40 femtoliters. So my device about 20 femtoliters, just 1.5 dilution or something. Thank you.